The Lion hypothesis is a hypothesis that explains gene dosage imbalance between males and females. One creative way of displaying male and female chromosomes is through these pictures, where I tape pins together to represent either the female, which is 2 X's, or the male, which is an X and a Y. Females have twice as many copies of all the genes on the X chromosome, but during the X inactivation process, instead of having double gene expression, that is cut in half, with one X being taken away. Once activated, all progeny cells have the same X as inactivated. The inactivated X is called the bar body, which can be depicted in the middle picture with the dime. They are inactivated, tightly compacted chromosomes that have very little protein gene expression, and they regulate the amount of X-linked gene product that's transcribed. So, to explain how X inactivation affects tortoiseshell cats and the type of phenotypes they get, we can look at the branch diagram below. As you can see, big B, and big Y equal the dominant color in cats. So there are 4 total possible gametes. If we were to mate a heterozygous male black long-haired cat with a yellow female short-haired cat, this is the results that we would get. As you can see, there's a 3/4 probability of having a yellow or black dominant colored cat, and also a 3/4 probability of getting dominant hair length, long hair. As you can see below, there's a 1/4 probability of having a recessive color, as well as a 1/4 probability of having short hair. If we were to multiply all these phenotypic ratios out, we would get a 9:3 to 3:1 ratio out of 16 possible opportunities. So because we had a heterozygous individual in our parent, this diagram depicts autosomal traits being passed down in our branch diagram. And you can see here are the 2 webkins that I found to display the different combinations of cats to produce a tortoiseshell cat. 2 female progeny from this cross would not have the exact same coat color pattern because male cats are typically either solid orange or yellow or solid black because they only have 1 X chromosome. But it is uncommon for female cats to receive 2 of the same alleles for fur color because they can receive 1 allele for 1 X from one parent and another color allele from another X for their other X on their other parent. That's how you get different crosses and tortoiseshell compositions in different cats, and that's why a lot of female tortoiseshell cats look different. It only happens for female cats because they have 2 X chromosomes. And here are my references. Thanks so much.